Okay, good morning to everybody. It's really a pleasure to introduce Irene Giardina. Well, because we are more or less the same age, so we start all this together. She got the PhD in Rome with Giorgio Parisi in 1997, if I remember correctly, because 8 is my year.
Yeah, but there is the usual delay.
Usual
when
delay,
you
okay.
get the piece of paper and when you actually finish.
Then she went to UK, then to France, and then she came back here in the Department of Physics. Now she's full professor here in the Physics Department. And she's leading a group which is doing something really world-leading, which analyzing this animal behavior in a way which has never been done before. So it's really something revolutionary in some sense, and everything started from this work together with Giorgio, one of the work of Giorgio Parisi that we decided to speak about. So it's really a pleasure to understand up to where they arrive with this kind of work, and so I leave the word to Irene. Thanks.
Thank you, Federico, for the nice introduction. It's really very nice to be here to talk about a paper that now is almost 15 years old. So it's a jump in the past, if you want, for me too. And actually, this paper had an enormous success and an enormous impact in, let's say, various disciplines. So what I would like to do is to try to explain you not only what we did and what the work is about, but also the context and why it had such an enormous impact in the following years. So as you can see from the title, this work is about animal collective behavior. And you already can see in my slide that in particular, the system that we worked on is related to flocking, and in particular to flocks of starlings, which are ubiquitous here in Rome. And actually, this was also the reason why we decided to focus on this system. Actually, collective behavior in biological systems is widespread in many species and on many scales. And here you can see in these slides several examples of this. You can see bacteria, aggregations of bacteria, and these are stem cells. So we are here on the scale of the macro, on the micro scale. But of course, there is a much more common, at least to our everyday experience, a kind of collective behavior, which is the one that we observe in larger organisms. So, for example, insects, fish, birds, and mammals. All these animals form aggregations, and these aggregations display collective patterns, and they can be either ordered or not, but anyway, there are these coherent phenomena which are common to our everyday experience. Of course, behavioral ecologists had noticed these collective phenomena since a long time, and their main aim was to try to understand why these individuals gather together to form groups, and whether there is a sort of evolutionary reason or a biological function related to grouping. On the other hand, starting from, let's say, the late 90s, collective animal behavior and collective behavior in biological systems started to attract the interest of different scientific communities. And why is that? The reason is related to the fact that in many of these examples, the collective behavior that we can observe is an emergent phenomenon. Namely, there are interactions between these individuals, and thanks to these interactions, a collective behavior at the group level emerges. So this mechanistic process of creating global patterns from local interactions is something that interests several different scientific disciplines. Physicists, of course, are very used to this kind of mechanism because it is what occurs in ordering phenomena in condensed matter. So when we think about ferromagnetism and phase transition, we are thinking about this very mechanism where local interaction gives rise to patterns on the large scale. But also, this is a problem which interests a lot engineers and control theorists. Precisely this kind of mechanism is what they call distributed coordination. Namely, it is a way to coordinate a group, but not from a top-down rule, but on the contrary, from a bottom-up mechanism. Okay, so they are very much interested in understanding what good mechanism for this distributed coordination could be. So in the end, these examples here, they represent very nice archetypes to look at when one wants to understand these different kinds of systems and different kinds of problems. Here I make some examples of the analogies between the living example. In this case, I chose a flock of birds, which will be the main topic of my talk, and the other examples from other topics and other disciplines. So here you can see two examples from the physics of condensed matter. I mentioned ferromagnets, but these are two examples which are even similar to the case of flocks. And why is that? This is, maybe you can't see it too well, but this is a dish with rice grains. And these rice grains, they are tapped. And due to the injection of this energy at the level of the individual grain, the whole aggregation starts to display alignment ordering. And the grains move together in a non-trivial way. These are experiments. I put the quotation, maybe it's too light and you can see it. These are experiments that were performed, I think, 10 years ago on rice grains. There are many other experiments. I just picked a couple of examples. And these are Janus particles. Also, in this case, these particles are activated by applying a magnetic field. And once they activated, they interact with each other and they display this collective motion, which is very similar to what observed in a flock of birds. Now, why I'm telling you that these examples are even more similar than a ferromagnet to the case of flocks, because in this case, each individual has a motor, okay? So each individual is self-propelled, exactly in the same way that a bird has a motor, has a self-propulsion mechanism, which is due to the fact that it eats and it transforms energy into motion. Okay, so these are examples from what is called the physics of active matter, where physicists study systems which are out of equilibrium when there is energy injection at the level of the single constituent. And here there are other examples from the social science and from robotics. Here you see a cartoon of a panic phenomenon where people, for example, has to escape for a very crowded area in a very short time, and there are these very crowded regions that are formed. This is also a problem which is related to the fact that there is a herding effect and many individuals try to act in the same way, precisely as in a flock. This is an example from mobile robotics. So these are some small robots which are controlled via some distributed rules, and they have to perform a task. Okay. And here, finally, there is a slightly different example, which is this is the chart of a financial market, where you see the price of some good, I don't remember which it was, but probably it is an index, this one, and you see these fluctuations and this non-trivial chart. And the price, if you consider it in terms of the action of the individual agents who put their bid, is again the product of the coordinating action of many individuals. So it is another, if you want, more sophisticated example of collective behavior in multi-interacting systems. Okay, so I wanted to show you this slide just to make you understand why when we started to work on this problem, there was this interdisciplinary interest on this kind of collective behavior, and people thought that there were analogies between these different topics, so that by studying one problem in detail, one could have inspiration and get inspiration also to address the other problems. So starting from these premises, in 2005, there was a European project, which was led by Georgia, which is called the Star Flag, okay? And there were several units across Europe involved in this project. There were groups of physicists studying models of collective motion in physics. There were a group of biologists. Here in Rome, there was the group of Giorgio Alleva, who were interested into the more biological aspects of of collective motion, and there were ornithologists in Germany, and then there were also economists. There was the group of Marcello De Cecco here in Italy, who was interested in macroeconomics, and the group of Jean-Philippe Bouchot in Paris, who was interested in financial markets. And so altogether, the idea was of, let's say, investigate this problem and exchange ideas about what we could understand of the mechanism of generative collective behavior in all these systems. In particular, our group in Rome was interested into really looking at the biological system, namely starling flocks. Okay, and indeed, the STAR flag is the acronym for starlings in flight. Okay. Good. So what was the system we were interested to, and why in particular flocks of birds were among the all possible examples considered a particularly interesting example? So this movie here that was taken by Claudio Carrere, who was a biologist. Now he's a professor at the University of Tuscia, but he was collaborating with us in that period. This movie here shows you very well that flocks of birds, they have this very coherent behavior. They are very nicely ordered. They move together. They are very cohesive, very coherent, and besides that, they are also very efficient in responding to attacks and to perturbations. So these are really some systems where the mechanism of moving together and when the collective behavior at the group level is efficient. So clearly, if you think, for example, at escape panic, that is a completely different example, where you have collective behavior, but in most cases, it is detrimental. So there should be something in the rules that lead to the collective behavior of the group, which might, in different cases, lead to something which is very efficient or to something which is not. And of course, people was very much interested to understand what this something is. So what are the features of the interaction between the individuals that make this group behaving in such a nice way? Okay, so this was the problem that we wanted to address. In the end, what we wanted to do was to learn the rule and to understand the mechanistic aspect of group formation and collective behavior, exactly as we would do in a physics system. So the difference is that in physics, we know what the interactions are, and typically we need to predict the macroscopic behavior. Here, it's exactly the opposite problem. We look at the macroscopic behavior and we want to understand what the underlying rules are. So it's what is called, from the statistical physics perspective, an inverse problem. Okay, so the big plan of our group was to learn these rules starting from the data. Namely, what we wanted to do was perform experiments and gather data on natural flocks of birds, and then look at this data, quantify the collective behavior, and then try to understand what were the rules giving rise to that collective behavior, and finally build models based on empirically motivated findings. Okay, so let me tell you a little bit what was the context when we started to address this problem. So the context, let me tell you first what was the context at the theoretical level. So biologists and computer graphics and control theorists and physicists have been doing models of flockings for a certain number of years, with different peculiarities related to the specific disciplines. So for example, the models devised by the biologists were typically with a lot of more parameters, okay? The ones developed by control theories, they were mathematically more rigorous, but even less reasonable, if you want, with improbable assumptions. And the ones of physicists, as it is in the spirit and the philosophy of physics, were minimum models, but with the idea of putting in these minimal models the ingredients that could reproduce the behavior on the large scale. So in the most famous model developed, and the minimal one developed by physicists, is what is called the Witschek model. So the Witschek model was published in '95, okay? And it just says the following. It says, okay, a flock is nothing else than a bunch of particles, but these particles are not like the standard particles that we are used to in physics, but they are self-propelled, they have a motor. So their speed is fixed. It is not determined by the thermal bath or whatever, they have a fixed speed, okay? And then on top of that, these particles can align with each other. And why alignment? Well, alignment because we know that this system develops collective order at large scale. This is what we want to model. And we know from physics that if we want to create a directional order on the large scale, we need alignment, okay? And by the way, this was a very reasonable assumption from what was known in biology. Since in biology, it was known that these groups display what they call allelomimetic behavior, namely each individual try to imitate the neighbors, okay? And in this case, imitation means to fly in the same direction. So it is an alignment mechanism. So the models capture in a minimal way all these ingredients, and it's just composed for each individual eye. There is an equation for the flight direction, the velocity, which tells us that the velocity of the next time step is the velocity of the previous time step plus an alignment term. So this is an alignment term. This is the average velocity of the neighbors. This k belonging to I are the neighbors of I, okay? And this means that a given individual I, look at its neighbors and try to fly in the same direction. So it's a very simple rule. And then the speed is fixed, as I was telling you, to model self-propulsion. And so we have basically the main ingredients of the model, which are alignment. Here we have the interaction neighborhood, which one has to specify, and then I'll tell you what it was in the Witschek model. We have noise, because decisions are always taken with a certain amount of uncertainty. And we have activity, we have self-propulsion. This is what makes the system of equilibrium. And then the position just moves in the same direction of the velocity itself, okay? So now an important point for what I want to tell you about this work. What are the neighbors that I look at when I have to decide where to move? So the Witschek model had a metric rule. So the idea was, I am the blue guy, I have to decide where to fly. So there is a metric range, okay? So distance, really a physical distance in meters. And I look at the neighbors within this distance. Okay, so that will correspond to this blue dotted circle in this picture here. So there is a metric range of interaction, which is what typically happens in physics. Okay, so you can take a sharp interaction range, you can take something that decays with a certain function, but the behavior would be more or less the same if it is short range. Okay, so this is so you have this bunch of particles that evolve according to this rule. And you can, for example, simulate them and you get some resulting behavior. And what is this behavior? This behavior is that, as in a ferromagnet, if the noise is low enough, the system develops long-range order and everybody moves in the same direction, okay? So it's a sort of off-equilibrium generalization of a ferromagnetic model. Why off equilibrium? Because the particle moves, so the directional degrees of freedom are not fixed on the sides of a lattice, okay, as in a ferromagnet. So they move, and the guys with whom you are interacting, they change in time precisely because of this movement, okay? So there is a changing, evolving in time in interaction neighborhood. Very good. So exactly as in physics, you can make individual particles models for a fluid, or you can look at mesoscopic theories and hydrodynamic theories. You can do also the same here. And people did that. So they just, what they did was, instead of looking at the individuals and the equations for the individuals, they took a small volume and they coarse-grained this volume. They got a density field and velocity field and wrote down hydrodynamic equations for this field. And so you get these field equations, these hydrodynamic equations, which are more complicated than standard hydrodynamic equations, because the system is out of equilibrium and certain conservation laws, which would be valid for a standard fluid, are not valid in this case. So the phenomenology is much richer. Okay, and again, this was in 95, 98. Okay, so these models, they are very reasonable. Okay, they capture in a very minimalistic way the idea that we have very reasonable idea of how these individuals should coordinate with each other, but they are completely speculative. I mean, there was no empirical test or experimental grounding of these models here. Okay, so I'm telling you this because in this way you have an idea of the general theoretical framework where we started to work. So what about experiments? I told you there were no experimental backings of these equations. Actually, there were experiments. So this was the state of the art in 2004 of experiments performed on natural groups. And you see what is the problem when comparing with the models. So these experiments were mainly performed by biologists, okay? And they were, let's say that the most accurate experiments were performed using stereoscopic photography, which I will tell you about. But what these researchers were able to do was to gather experiments of very small groups, small of a few tenths of individuals, okay? So now when you have a few tenths of individuals, you cannot perform any sort of statistical quantitative analysis. If you think at a group of 20 individuals, I mean, everything is on the surface. You don't have the bulk. So we know in physics, everything is about the bulk. Everything is about large numbers, especially in statistical physics. So there was no hope with this data of trying to compare with the models and try to understand whether what were the mechanisms giving rise to collective phenomena. No collective phenomena with 20 individuals. You need to have larger numbers. So we wanted to gather data with larger numbers. Okay. Okay, so the experiment, so we started doing experiments. We knew nothing about the stereoscopy, nothing about birds, nothing at all. So this was completely new. And we were actually very naive. We thought, okay, we are the physicists, we can do this in 2 days. Actually, we spent 3 years doing experiments and tried to understand what we were doing. Okay, so experiments were performed using stereoscopic photography. What is stereoscopic photography? Stereoscopic photography means that you take multiple images of the same object from different observation points. And I will explain you why it is useful in this case. But first, I wanted to show you where we did the experiments. We did the experiments on the roof of a museum of Palazzo Massimo at the railway termini station. And why is that? Because during winter, the square in front of the railway station is a roosting site for starlings. So every day, I mean, around 4 o'clock, 5 o'clock in the afternoon, all these flocks come back to the roost and they perform this flocking behavior above the trees before landing for the night. So it was an ideal situation where we could shoot at them. Okay, because they were on the roof, we were close enough to the groups to try to have the necessary sensitivity on our images to be able to capture the single individuals. And there were many flocks, so we could hope to gather a good statistics. Okay, so this is what is stereoscopy. What is stereoscopy is based on some stereometric relationships, and the idea is the following. Let's say that we want to understand how this guy is behaving. Understanding how this guy is behaving when you think at a system that is moving means that you want to retrieve the 3-dimensional coordinates and velocities of this individual here. Okay, so let's start from the coordinates. If you want to retrieve its coordinates and you take just one image of this guy, you will have a 2-dimensional projection. But what we wanted were the 3-dimensional coordinates. So to do that, you exploit the fact that if you take 2 different images, let's say that these are the 2 CCDs of our 2 cameras, okay? And so we take 2 images of this individual from different locations. The projection on this image of this guy, and the projection on this image of this guy will be different. So here you see, I called it XA, and here I called it XP. So there is a difference in the coordinates on the 2 images, which is called the stereoscopic shift in stereometry. And how much is this stereoscopic shift? Well, it depends on the distance of the object and on some geometrical features of your setup. The distance between the 2 cameras, which is this D, and the focal length of the lens that we are using, okay? So basically, the idea is that if we can measure this disparity, taking multiple images, okay, we can measure the focal length and the distance between the two observation points. And exploiting these formulas, we can retrieve the missing coordinate, which is the z. Okay, so the conceptual idea is very simple. Of course, everything becomes much more complicated because we don't know d with infinite accuracy. We don't know the focal length with infinite accuracy, and so on. There are many geometrical coefficients of our apparatus that we don't know exactly. We know them only up to a certain precision, and this makes things much more complicated. So if you make a little bit of computations and you know that your flocks from the roof will be approximately at least 100 meters from you, and you know that the minimal resolution, the best you can do is to discriminate the points with a difference of 1 pixel, which is the discrete minimal feature that you have on your CCD. And if we want an error, which is of the order of centimeters on the relative distance between individuals, you can make the computation, make all this, put all these numbers in these formulas. What you get is that you have to take the distance between the two cameras larger than 20 meters, okay? And this is why we went on a roof, and we needed a very large roof because we needed to put these cameras very far away one from the other. And now to tell you, to try to understand why eventually we did much better than all the other groups before, you have to consider that, I mean, other experiments were performed with a baseline of 5 meters rather than 25, which is what we used. But actually, this is just part of the problem. So this is actually the actual setup that we used. So from what I told you before, you can understand that the more observation point you have, the more is the chance that you recover the missing coordinate with a good precision. Okay, so in what we actually did was to use three observation points, 2 observation points close one to the other, and the third one far from the first 2. At the time, you have to think that now in my group we perform experiments. We still perform experiments of this kind, but we have cameras that shoot at 175 frames per second. Okay, and with a very good resolution in terms of pixel. In 2005, what we used were these cameras which had a resolution of 8 frames per second. So they were much slower than the cameras that we have today. Okay, and why is that? And the reason is that there is always a trade-off between the resolution in pixel of the CCD and the frame rate at which the camera shoots, because the amount of information that you can record on your memory, and so that you can just transmit to your recording device per unit time is more or less the same. So you have to bargain between these two. Now it is better for technical reasons, but at the time we had to choose between a large resolution and a large or a larger frame rate. And at the beginning we chose the resolution, and we did well actually, because in that context it was a good choice. So these were the cameras that we used. This was the apparatus. The cameras were convergent. They were aligned in a very creative way devised by Andrea Cavagna, who worked in this project with me and George and other people. And to increase the frame rate, we also built a trigger and a temporization device. And actually, Nicola Cabibbo built this device. I don't remember, maybe Giorgio remembers this. And it was a very nice temporization instrument. Okay, so this was our setup, experimental setup. Now, what is the problem in the 3D reconstruction of flocks or other animal groups? The problem, you can grasp it from these images. So stereometry allows you to do the following. Once you know that your object has a certain image in one camera and another image in the other camera, you can exploit the formulas and retrieve the full 3D coordinate. But to do that, you need to know who is who, namely, which bird on the first image corresponds to the same bird on the other image, okay? And when you have one object or two objects or 10 objects, this is simple. When you have 2,000 of them, this becomes extremely complicated. So for example, in this case, it's easy for you to tell that this guy is this guy, or that this guy is this guy. But when you go inside the flock, it becomes much more complicated. Okay, so this is known in stereometry as the matching problem. It's a well-known computer vision problem. Okay, so this guy here, then it turns out to be the match of this one, but you wouldn't say that if I didn't tell you. Okay, so it becomes, this matching problem is really very, very complicated. And this is why other people didn't know how to do that. Okay, and biologists didn't know how to do that because they were not used to this kind of technical problems. But even people who were used people in computer vision who were used to perform this kind of tasks, they were not able to solve it. And why is that? The problem is related to the numbers and to noise and to the presence of noise. Okay, so usually what happens is that given a bird, an image on one camera, stereometry tells you that this bird here corresponds to a line on the other image. So you know that the match must lie on that line, okay? Fair. Then if you have few individuals, it is easy to do that. You just compute what is called epipolar line. You have just one guy that crosses the epipolar line, you do the match, okay? But when there is noise in your problem, you will never find this guy which is exactly on this epipolar line, because the epipolar line is not computed in a very precise way. So this blue guy will be a little bit above, for example. And if there are, instead of 1,000 individuals, you have a large chance to find another guy who is not the correct match, who is as close to the epipolar line as to the real match. So it's really a matching problem, okay? And so you need to find a way to perform this matching task. And what we did was to build a measure, okay, and to perform an optimization problem. Once you build a measure, which tells you how likely it is that you can find a match, you then optimize this super matricial matrix, and you find, as you would do in statistical physics, in any statistical physics matching problem. So we needed to build a measure, and the measure is built knowing stereometric relationships, but this is not in general enough. What we did at the time was to implement, to look for patterns. Namely, we just not required a potential pair of matches to satisfy well enough the epipolar constraints, but we also required that patterns around that individuals satisfy the constraint. Okay, and using this pattern mechanism, we were able to solve the problem and to perform the 3D reconstruction quite well. And so here you see one of the largest flocks that we reconstructed at the time. So here are the two faraway images at 25 meters, one from the other. And this is the real 3D reconstruction. So actually, this is the same perspective of the image, but this we are rotating it because we have the full 3D coordinates. So we got all these 3D coordinates, and this is almost 3,000 individuals. So it's 2 orders of magnitude as compared to what people did before. Okay, so this was a very strong improvement from the point of view of the experimental data and of the consequent statistics that we could perform on the data. Okay. Now, fair enough. So once we had all these 3-dimensional coordinates, we could do a lot of things. So today I'm focusing on the results discussed in this paper, which was our first big result. And what we did is the following. We computed all the standard quantities that you would compute in a particle system in a liquid, say, but we didn't find anything particularly interesting. But at some point we said, okay, let's see how individuals are distributed around a given focal individual. So let's say I am a bird and I want to understand where is, for example, my first neighbor. Is my first neighbor equally likely all around me, or there are privileged directions? Okay. So let's say this is the guy, this is me, and I want to look at my first neighbor. So my first neighbor, for example, is here. Since we are in 3D, the angular distribution of my first neighbor is specified by 2 angles. So it's specified on a sphere, if you want, a sphere centered in the focal individual, okay? And I want to compute the distribution on this sphere. Okay, so we computed this distribution, and to visualize it, it's better to open up the sphere. This is what is called the Moll-Wade representation of a sphere. And you see this Moll-Wade representation here. So the focal individual is in the center, and the axis which goes from the slide outside is the front of the bird, okay? While these 2 points correspond to the back. I'm opening up the sphere. Okay, and the color map stands for yellow corresponds to a lot of neighbors, a high probability of finding the first neighbor, and the dark red corresponds to very low probability of finding the first neighbor. Okay, so this map here is telling me that when I look at the distribution of the angular position of the first neighbor, it is much more likely to find the neighbor on the sides rather than in front or below of a given guy, okay? And if you think about it, this anisotropy in the distribution of the first neighbor must be due to the interaction between individuals, okay? And this is also very reasonable. When birds fly together, they don't want to bump, for example, with the guy in front, so they tend to keep a larger distance. So it is more likely that the neighbors will be on the sides. This is a possible reason. Or if you look at what is what are the ocular features of birds, you discover that the optical axis of the eyes of the birds is lateral, so they see better on the sides. And that's why they position their neighbors on the sides, probably because they are able to track them in a better way, okay? So there are simple and very reasonable explanations for why we find this kind of anisotropy, but the important point is that this anisotropy must be due to the fact that there is an interaction between the individuals. And so this means that we can use this anisotropy as a proxy for the interaction itself, okay? Now, what I've been telling you is for the first neighbor, but we can repeat exactly the same kind of analysis for the second neighbor, the third neighbor, and so on. So here is the same map, but for the 10th neighbor, okay? So you see that for the 10th neighbor, the distribution is basically isotropic around the individual itself. So this means that if we use this anisotropy as a measure of the interaction, the guy is interacting strongly with its first neighbors, but then this interaction decays as we consider farther and farther neighbors. Okay, so we would like to quantify this qualitative result in a sharper way, and so we do the following. We introduce this matrix here, which is a neighbor projection operator. This is built in this way. What I do is that assume that this is the guy, the focal guy, and I want to study the distribution of neighbors around it. And this is the first neighbor. So this red guy is the first neighbor. I just build a vector that goes from I to the first neighbor. I call this UI, okay? UI1, because this is the first neighbor, okay? And then I can do that with the second neighbor, the third neighbor, and so on, okay? Then let's say that I consider the first neighbor for this time. Then with the first neighbor, you have to put 1 here. This index stands for the order of the neighbor. With this first neighbor, I build this matrix here. And this matrix, if you look at it, you recognize that this is the structure of a projector. So this is a matrix that projects along the direction of the first neighbor, but it is averaged over all the individuals. So for each individual, I compute the location of the first neighbor, I draw the vector, I build the projector, but then I average over all the individuals in the group, okay? This is the average neighbor projector matrix. And then what I do is that I compute the eigenvectors of this matrix and the eigenvalues of this matrix. And by construction, you can do, I mean, in a piece of paper in 2 seconds, you can do the computation. By construction, you can see that the smallest eigenvalue, the eigenvector corresponds to the smallest eigenvalue of this matrix, defines the direction of minimal crowding, okay? The direction along which there are less neighbors, okay? So if I take this vector here, this eigenvector, it is a vector, I can take the scalar product with the direction of motion of the birds, okay? And this scalar product should be large if the direction of minimal crowding is in front, as we saw before from the map, and it should be small if it is not. Okay, and in particular, if the distribution is isotropic, this scalar product should be equal to 1/3, because this is statistically the average value of the angle between two random vectors. Okay, so this is a quantity that is large if the minimal crowding is along the direction of motion of the whole group, and goes to 1/3 if it is random. Okay, and so we can compute this scalar product at this point is just a function of the order of the neighbor, because I can repeat the scalar product for the first, the second, the third, and so on. And I can plot it as a function of the order of the neighbor. So this is the ones that I presented you before is an anisotropy factor. It tells me how strongly anisotropic is the distribution of neighbors, and therefore how strong is the interaction between individuals. So we can use this anisotropy or interaction strength and plot it as a function of the order of the neighbor, and we get something like this. This is for two different flocks. So you see that, as you expect, also from looking at the maps, okay, this interaction strength, which corresponds to the degree of anisotropy, decreases with distance. Okay, so at some point it reaches the value, the non-interacting value, and we can take this point as a measure of the interaction range. Okay, so for these two flocks here, this interaction range is around here, so it is around 7 individuals. Okay, of course, here I'm plotting this strength of interaction as a function of the order of the neighbor, but I could have done exactly the same procedure, but not with the order of the neighbor, but with their distance, their physical distance. And in that case, I would get exactly the same plot, but here on the x-axis, I would not have order of the neighbor, but I would have distance, okay? And in this way, I can compute also a range in terms of distances. So after looking at this function here, so I can compute the interaction range in terms of number of interacting individuals, which is what I call the topological range of interaction. And I can compute the interaction range in metric distance, which I call the metric interaction range. Okay? Okay, that's good. So for one flock, I mean, I can choose either one or the other to quantify the interaction strength in my single flock. Indeed, there is a relationship between the two, okay? And what is this relationship? If your system is homogeneous, okay, let's say that you want to compute what is the relationship between a neighbor of order n and its distance. A very simple argument is, let's build a sphere, okay, around the focal individual. And the radius of this sphere should be such that at the given density of that flock, NC individuals are inside that sphere, okay? So this is just a homogeneity argument, which can, in any case, can be proven by measuring that relationship in the data, and it holds. But this tells me that the relationship between the topological interaction range and the metric interaction range is this one. Okay, is this one here. And you can express it in terms of the density of the flock, or since this power of the density is proportional to the nearest neighbor density or sparseness, you can equivalently express it in terms of the sparsity, R1. Okay, well, that's fine. For one flock, rho is given, R1 is given, so one range is equivalent to the other. Okay, but what happens when I look at different flocks? So when I look at different flocks, the first thing that I notice is that they have rather different densities. So the density of a flock changes from flock to flock, okay? So this quantity here changes, okay? Therefore, you cannot have this and this always the same in all the flocks. So there are two possible scenarios. One scenario, let's say a la Wieczek, the model that I presented you before. What Wieczek said was, okay, there is a fixed metric range of interaction, okay? So in that case, RC should be fixed in all the flocks, okay? But then, since RC is fixed in all the flocks, ANSI should vary from flock to flock because this quantity varies. Okay. The other possible scenario is the complementary one, that NC is fixed in all the flocks. And in that case, RC should change from flock to flock. Okay. So we had data on many different flocks, and we could check which one of these two scenarios was the correct one. Okay, and so this is the result. So what you can see is that if I change the density of the flock or sparsity, it's convenient to compute the sparsity, but this is a measure of density, so it's the same. So if you put on this axis the sparsity of the flock, and you look at the topological range of interaction, NC to the minus 1/3, and we take this power because it is the one that it's equivalent to RC in the relationship between the two. Okay, so if you look at this NC, it fluctuates but around a constant. Okay, on the contrary, if you look at RC, it increases linearly. Okay. So this is not what should happen in the Wieczek scenario. In the Wieczek scenario, we will need to find a constant here, data fluctuating around a constant. So it's not in real flocks, it's not like in the Wieczek model that there is a fixed interaction range in physical distances. It is a different situation where there is a fixed interaction range, but in terms of number of interacting neighbors. Okay. So what does it mean? And this number, by the way, is approximately 7. So this means that if you have a very dense flock or a very sparse flock, the yellow guy always interacts with the first 7 neighbors. So the degree of the degree of interaction in terms of number of interacting neighbors is always the same in sparse flocks and in dense flocks. Okay. And you can immediately understand that this is very convenient because any kind of disturbance which alters the density of the flock does not change the degree of local interaction. So you can already understand that from this simple argument that this kind of we call it topological interaction should be more, let's say, more effective to maintain the cohesion of the group. Okay. So this is what basically what is meant in the title of the paper. Interaction ruling animal collective behavior is topological and not metric. This is exactly what is meant in the title. Okay. So so now why? So this kind of interaction, this topological interaction, is very different from what is found in physical systems. In physics, all the interactions depends on the distance in meters. So it's very different. So what could it be that generates this kind of interaction? Well, in the period where we were finding these results, by chance one day we met some biologists. They were neuroscientists and biology. I don't remember exactly when where we were. It was a conference, I don't remember exactly, but at some point we discussed with them this thing, and they pointed us to some interesting literature where people had been studying cognitive abilities in animals. Okay, and they had performed a whole series of analyses on different species. Okay. And they were interested in quantifying what they called the pre-numeric abilities of animals, which does not mean that animals know how to count, but it means that they are able to perform pre-numeric tasks. So, for example, in these famous papers, what people did was to try to understand whether birds could discriminate different numerosities. So these birds were exposed to groups of different delicious things, some less and some more, and they had to choose between them. And in this way, in a quantitative way, they understood what was the maximal numerosity that birds and other species, they performed on different species, these experiments were able to discriminate. Okay, and this is the paper where they looked at birds, and what they found out was that the largest numerosity that the birds were able to discriminate is around 7. Okay, so if you present a bird with a group of 3 very nice grains and 6 very nice grains, they pick up the larger one. Okay, but when you present them with two dishes, one with 9 grains and the other with 10 grains, they perform a lot of error in discrimination. Okay, so this 7 look like a bottleneck for this discrimination task. But there were other experiments that were focusing on what is called a subbatizing ability of animals, which means the ability to track simultaneously a certain number of objects, which when you fly, it's pretty important. Okay, and again, it was found that the maximum number that a bird was able to track at the same time was around 7. So the explanation, I mean, the interpretation in connection with our results of all these findings is that, okay, this number of interacting neighbors is around 7 because this is the maximum number of individuals that they can keep track of while they fly. So there is a cognitive origin because of for these strange kind of interactions, which clearly you cannot find in physical systems. But once you start looking at organisms which are more sophisticated, which have a higher degree of abilities, you might find. Okay. And now we also wanted to check a little bit the idea that I told you before, that this kind of interaction grants more robust cohesion to the group. Okay, and to do that, we performed numerical experiments, numerical simulations. So we simulated the Witschek model, but the Witschek model with 2 different rules. In one rule, the interacting neighbors were chosen as Witschek did, with a metric rule. Okay, and in the other case, they were simulated with a topological rule. And then once we simulated these two flocks, we make them crash against a perturbation. Okay, and so let's say that we send the flock against an obstacle, namely something with a repulsive interaction, and we just measure quantify the effect of this repulsive interaction. And you can quantify this effect by counting the number of distinct groups in which your starting group is split into. So let's assume that there is a flock. This flock goes against a falcon and it splits. Okay, it wants to avoid the falcon, and to do that, it splits. And it can split in 1, 2, 3, several subflocks. And the number of these subflocks is a measure of the cohesion of the group. So what you see is that in the metric case, the number of subgroups is around 5, 6, 7. I mean, you have this wide distribution. While on the contrary, for a topological flock, the most part of the time, the flock manages to remain all together. So it's true that cohesion is more robust when you use a topological rule. Okay, so this is a small summary. I wanted to, my last slides are a little bit about the aftermath of this paper. So the summary is that interaction between individuals are topological, not metric. The nature of interactions is related to the cognitive ability of individuals. Topological interactions grant more robust cohesion. And topological interactions are different from what assumed in models. So this is a very concise summary of what we have been discussing so far. So just a few words about a further validation of these results that we obtained in 2005. They were published in 2008, but we took 3 years to get them. Okay. We did it with several collaborations. We did in the following years. So we tried to measure this interaction range using completely different methods. Okay. So in a collaboration with some colleagues of ours in Princeton, we said, okay, let's apply some standard statistical physics techniques to infer from data on the velocity correlation, so other quantities completely, the interaction range. It's very long to explain how we did, but the important thing is that it's a completely different method with completely different kind of data. Okay. And we got the same results. So you see these two plots are the equivalent of the ones I've showed you before topological range versus sparseness, metric range versus sparseness, you find the same behavior, okay? So this is a confirmation with a completely different technique of the result that I've been presenting you. So we did more simulations to prove this robustness in cohesion, okay? The first simulations were in 3D, these ones were in 3D. I mean, we just proved again the same feature, namely that topological interactions are more robust, okay? And then this, I don't have time to tell you what is this, so I will skip it. Okay, so I've been telling at the beginning of my talk, I've been telling you the connection between the world of collective behavior in biological systems and in animal groups with a lot of different communities. And I wanted to do that, even though I spent a little bit of time, because this also explains the impact, the enormous impact that this paper had. Okay, so today, I just checked this morning, the citations of this paper are more than 2,000. So it's really a highly quoted paper. A lot of people read it, okay? And why is that? Precisely because the mechanistic aspect of group formation is shared by many different disciplines, and this is really an archetypical case, which is very easily, if you want, reachable from different communities. And to show you that, I just wrote down the 3 last citations to this paper. So these are very recent citations, the last 3 that I found. And the first one is, I read you the title. It's something about tissues. So these are physicists. Ricardo Aredi is a physicist, a group of physicists that try to understand how an epithelial tissue works. Okay, and they use techniques from the statistical physics of active matter to do that. And they cite our paper because it's one of the first paper where in an active matter system we measure the interaction rate. Then this other paper is apparently completely different. Modeling the pedestrian flow before bottleneck through learning-based method. So these are people who study traffic flows, pedestrian flows. So it seems something completely distant, but actually it is not, because again, herding mechanisms are mechanisms where you have imitation, you have people working together. And so there is a connection also with what happens in collective animal behavior. And the third paper, nonlinear pinning control of stochastic network systems. This is people from control theory. So these are mathematical engineers just study algorithms to control robotic systems. Okay, and so this gives you an idea of the wide impact that this work had. Okay, so just 2 words more in particular in physics. So in physics, what was the impact of this work in physics? I mentioned the Vitsek model. So the Vitsek model actually had per se a lot of relevance in the context of the statistical physics of out of equilibrium physics and of ordering phenomena. And why is that? Because I told you that this model is very similar to ferromagnetic systems, but it has a peculiarity. A ferromagnet in 2D does not order, while the Vitsek model in 2D orders. So this model opened up a completely new class of kinetic ordering phenomena, which are intrinsically out of equilibrium. So people studied a lot these systems, and there was a huge debate that lasted until, let's say, 5 years ago about the order of the transition. And eventually people understood, I mean, they agreed that the transition was first order, okay? And it was first order, there were instabilities, there were bands around the transition point. So when you consider a flock before periodic boundary condition, flock with periodic boundary condition, before the ordering, it starts to develop traveling bands. Okay, so this is an instability and it's a signature of first order transition. This is with a metric rule. But what happens if you put a topological rule? So once we published this paper, people started to look at this, okay? And what they discovered is that with the topological rule, the transition becomes a second order, okay? So this gives rise to a lot of interesting things that one can do in kinetic theory, hydrodynamic theory, and so on, okay? So this is about physics. Now, what is the broad interdisciplinary impact of the paper and of the work. Okay, at the level of if you want, the study and research on living system, and in particular on animal groups, we showed for the first time that it is possible to perform experiments on the large scale on big animals, okay? But to do that, you need to have the skills that a physicist have. Because in the end, to perform the 3D reconstruction, we exploited our background as physicists. We built a measure and we solved an optimization problem. A biologist would not have done that, and a computer vision scientist would have not done that. So it was precisely our background as physicists that allowed us to solve this problem. I mean, every problem where you have noise, typically physicists can give their contribution. So people understood that. So once we went around, we explained our results and so on, people really started to understand this. And from that moment on, a very large number of groups of biologists studying collective behavior started hiring physicists. So, for example, here I make it. I just mentioned the three groups: the groups of Guy Taylor, who is a very famous guy working especially on ants. They did some very nice work on ants. They started hiring physicists. Ian Cousin, who is now he's the head of the Max Planck Institute for Ornithology in Constance, and I mean, he has started hiring physicists. So I mean, people just really recognize that there is a perspective, there are some technical skills that physicists have. But this is not the most important point. The most important point is that we also try to give the idea that you need to have a change in perspective. It's not that if you look at 10 individuals, it's the same as looking at 2,000 individuals. Okay, big is different. Okay, so if you really want to give a statistical description of large-scale behavior, you need to look at large numbers, and you need to look some approaches based on statistical physics. And also, there's this message passed through the community. And now, I mean, at the beginning, it was very hard. We took a long time to have this paper published. People didn't want to publish it because it was too far from what biologists were used to. Okay, but once it was published and people really understood what it was about, things changed. And from our point of view, this also, I think, opened up a new perspective and a new approach to the physics of living system, which had a very long tradition at the microscopic level. There was a lot of people working on cells, working on tissues, working on bacteria, but no one worked on starling flocks, on fish schools, on larger scale organisms. But it is possible. It is possible, but you always need to take into account that even though you want to apply the same methods and the same approach as in physics, minimum models, you need to be open to change the rules, because the rules might be different, just because these are not particles, these are organisms. Okay. And then the last slide, which is sort of, I tried to look for some images, but I didn't find many. So these are the authors of this paper. Some of them are in the audience, okay? So there was Andrea Cavagna, this is me, this is Giorgio. These are all the other people that work with us in this in this work. And this is the only image that I found. So this is at the beginning of the project, where it's an image taken at Circo Massimo, and we wanted to test our stereometric setup, but we needed a very, very big place. Okay, and so the only place that came to mind were Circo Massimo and Piazza di Siena, but then eventually we chose Circo Massimo. And so here you see this red triangle. Actually, I could zoom in this red triangle and you would find the 3 small targets. So these tests were some tests that we did with our apparatus very far from the targets to try to retrieve the 3-dimensional coordinates of the targets and of their distances and see whether we achieved the good enough accuracy. So these were just some tests at the beginning. So this is me, this is Andrea Cavagna, this is Ballerini. Okay, so he was in charge of all the electronics. Andrea Procaccini, who was a PhD student working with us, and Vladimir Zrakovic, who was a postdoc in our group. And unfortunately, the other ones, I didn't find an image with all the other ones, but everyone worked on a different aspect of the work, and that's it. Thank you.
Comments?
Well, thank you so much for this very interesting and very accurate recollection. One thing that I think that you have not mentioned, but I think that you may briefly mention, is the fact that one can do some analytic computation for flocks in higher dimension and so on. Which is just to say that you can do something on that direction.
Yes, I mean, you can do analytical computations. I just wanted to focus on the topic of the paper, but then starting from these first results, then if you remember, in one of the first slides, there was the plan. And the last point of the plan was to do models. Then we also did the models, but that came later. Okay.
Any other question? Actually, I have one question.
Okay.
I suppose that on the boundary of the, there are differences. So
yes.
When you take the analysis, you say, we take the average over all the birds. You mean only the central ones? You have to be careful with the borders.
The question of a physicist. Okay?
Exactly.
No, of course.
I use periodic boundary condition.
You need to take care very carefully of the boundary. So otherwise, the anisotropy is completely screwed up by individuals on the boundary. This has been done. Actually, this was a problem with all the statistical analysis performed by biologists in the past, because they did not take into account boundary effects at all. So for example, they had all these papers telling that there was a pattern in the density, that the density was larger or smaller. The density decreases as you increase the size. Oh, it's just that it's the effect of the boundary that, I mean, decreases as you increase the size. So there were all these problems with the boundary, but this has been taken care of here. All the data are detrended by the boundary effect. Yes.
Thanks. Any other question? Otherwise, we thank again Irene for the nice talk. Okay, so now we have a coffee break outside, and at 4 o'clock we reconvene for 4 and 5 minutes for the second seminar. Thanks.